this first A-level biology video about proteins, we're going to look at the structure of an amino acid and the 20 different amino acids, and how these can be joined together by condensation reactions to form a peptide sequence. And then we're also going to look at the different levels of protein structures, so primary, secondary, tertiary, and quaternary proteins. You hopefully already have some knowledge about proteins from your GCSE studies, so before we dive into new content, just pause the video and scribble down what you already know. Hopefully you're aware that proteins are synthesised by the ribosomes, and they're made from amino acids. Proteins are used in the human body for structural fibres in muscles, for making hormones, and also for making enzymes, and these are all functions that you would have studied as part of GCSE biology. Now, proteins are actually a really diverse group of polymers made from long chains of amino acids. And we can break down their functions into four main groups. So we have structural proteins, like the ones that make up muscles and ligaments and hair. We have catalytic proteins, because of course enzymes are made from protein, and enzymes are responsible not just for things like digestion, but really for speeding up all of the chemical reactions that happen in a cell. We've got signalling proteins, both as hormones and also as receptors, and then also immunological ones. So antibodies are also made from protein. There are 20 naturally occurring amino acids, and we can split these into three groups based on whether it's vital that they're included as part of the diet or whether it's possible for the body to make them from other amino acids. So we start off with 9 essential amino acids, and these are the ones that cannot be synthesized from other ones. So it's really important that they're part of your diet, and that's particularly vital if maybe you're a vegetarian or restricting your protein intake for some other reason like an allergy, and it's really important that you're actually getting all 9 of these essential amino acids. Then there are 5 amino acids which are non-essential, and it's possible to make them from other amino acids. And then there are 6 which we call conditionally essential. So these are ones that in most circumstances a healthy adult human will be able to make from other amino acids, but a baby or child probably won't be able to. And also an adult human may not be able to, either if they're under large amounts of stress, or maybe if they're injured or involved in endurance exercise, in which case it may not be possible for the body to convert enough amino acids to make these ones, and they would need to be part of the diet. All 20 amino acids follow the same basic structure, and here I've drawn glycine, which is the simplest of the amino acids, just so we can look at the general structure. So in the centre of the molecule we have a carbon atom, and carbon, as we know, can form 4 covalent bonds. So one of these covalent bonds is to the carboxyl terminal. So this is basically the functional group of a carboxylic acid. Opposite that we have the amine terminal, and watch out for the fact that this is amine, not amino. So that amine terminal consists of a nitrogen atom covalently bonded to 2 hydrogen atoms. Now in glycine, our remaining 2 bonds are both to hydrogen atoms. All amino acids are going to have 1 hydrogen that's bonded, and it doesn't matter whether you do that above or below. And then they're also going to have what we call an R group. So in glycine, the R group is also a hydrogen atom, and that makes it incredibly simple. But as we look at some more complicated amino acids, you'll see that the R group can actually be quite involved. You're not expected to memorise the 20 amino acids and their R groups, but you could be given information about them and then asked to draw conclusions, so it's worth having a little look at them anyway. The first 6 are the non-polar aliphatic amino acids. So they're non-polar because there's no oxygen involved in them, so we don't have any of those polar bonds. And that's important because when proteins start folding up and developing a 3D structure, one of the ways that this can happen is the formation of hydrogen bonds between different polar amino acids. So the fact that these are non-polar means that they won't be forming those hydrogen bonds. You can see that the majority of them are just hydrocarbon chains, with the exception of methionine. Now you'll notice that that has sulfur in, and I have highlighted it in yellow because sulfur is yellow. But when we start talking about disulfide bridges, it's important to note that methionine can't form disulfide bridges, even though it has sulfur in it. Next, we have our polar uncharged R groups. So you can see that serine and threonine have got this oxygen in there. You'll also note that cysteine is another one of these amino acids with sulfur in, and it's the cysteine that forms the disulfide bridges, which we'll talk a little bit about in a second. Now, proline looks like a really straightforward, simple hydrocarbon chain, but you'll also notice that it has this one random bond coming out at the end, and that's because the proline R group doesn't just sort of stay on its own. It does this quite distinctive thing where it forms a cyclical structure and it loops back round and joins onto the amine terminal, which makes proline very strange. And then we've also got asparagine and glutamine, which have oxygen in them, and this is what's giving us this polarity. Phenylalanine, tyrosine and tryptophan are described as being aromatic, so that's in contrast to the first ones we looked at, which were aliphatic. So aromatic means they have this benzene ring, which is this sort of hexagon with additional lines in it. And then finally we have our fully charged amino acids. So these are not just slightly polar, they're fully charged. So lysine, arginine and histidine are positively charged, and then aspartate and glutamate are negatively charged. Before we go any further, pause the video and make sure you can answer these questions about what we've covered so far. The basic structure of an amino acid is a carbon atom in the centre with the amine terminal on one side, so that's another 3 atoms, and the carboxyl terminal on the other side, so that's another 4 atoms, with a hydrogen atom, that's 1 atom, and then the R group. Here we're told that glycine's R group is a single hydrogen atom, so if we add all of those up, we have a total of 10 atoms. You should be able to draw the structure for glycine just as we described, and then the molecular formula for glycine is going to be C2NO2H5. Now you may have already encountered in some places that it's appropriate to write out a formula in a way that kind of explains the structure. So for instance, writing COOH. But this question has asked us for the molecular formula and therefore we're not expected to do that. We should join together all of the atoms of the same type. The structure of an amino acid includes 1 R group and 2 other groups. Name these 2 other groups. So we've got the amine group, and it's really important that you're calling it amine, not amino, and then also the carboxyl group. A scientist recorded all of the elements in an amino acid. Which elements did they record? Well, our basic structure for an amino acid looks something like this. So we can see that regardless of what the amino acid is, it's going to include carbon, oxygen, hydrogen, and nitrogen. So therefore we can rule out option B because it doesn't include oxygen. Then we know, having looked through all of those previous amino acids, that none of their R groups contain phosphorus. So phosphorus might be on the brain for you because when we talk about the 6 most abundant elements in living things, phosphorus is in there because it's necessary for ATP and for DNA, but it's not necessary for proteins. So neither of those is correct either. So therefore our correct answer is going to be D. A slightly larger molecule called a dipeptide can be made when 2 amino acids join together. This is another example of a condensation reaction, and a water molecule is lost. The bond that forms between these is called a peptide bond. When we start to join together more than 2 amino acids, rather than making a dipeptide, we can make a polypeptide. Just as we saw for carbohydrates, a condensation reaction can be undone by a hydrolysis reaction. So lysis means splitting and hydro means using water. So we can take our dipeptide or our polypeptide, and by introducing water across the peptide bond, this can be broken to take us back to our 2 amino acids. So 2 amino acids can make a dipeptide and multiple amino acids can make a polypeptide. But to get to a protein, we need more than just a sequence of amino acids. We need these to be folded into a 3D shape. When we start thinking about protein structure, there are 4 different stages we go through: primary, secondary, tertiary, and quaternary. The primary structure of the protein is simply the sequence of amino acids that have been joined together, and this is determined by the DNA code. So when you look at a strand of DNA, we can break every 3 bases into what's called a codon, and each one of those triplet codons codes for 1 amino acid. So when the ribosomes then assemble those amino acids and join them together, we have our primary structure. Once we have this linear polypeptide, so that's the primary structure of the protein, the next thing that's going to happen is that hydrogen bonds are going to lead this to have a secondary structure. So the hydrogen bonds are forming between the slightly positive amine end of one amino acid and the slightly negative carboxyl end of another amino acid. And there are 2 possible options for what this might do to the protein. It could either coil it into an alpha helix, or it could fold it into a beta pleated sheet. You already know the word helix from using it to describe the shape of DNA, which is a double helix. So a single helix is just a spiral, but it's one with a continuous diameter, so it's not getting wider as it goes. And rhodopsin is a classic example of a protein where its secondary structure is an alpha helix. So if we think about how this alpha helix forms, we have a whole bunch of amino acids here, and they're joined together by these peptide bonds, these strong covalent bonds between one amino acid and the next one. And then, as we know, the oxygens of these carboxyl groups are going to become slightly negatively charged, and the hydrogens of the amine terminals are going to become slightly positively charged. And this allows a hydrogen bond to form between these. So remember, this isn't a covalent bond, it's not a strong bond, it's a weak intermolecular force. But this is what is causing this long strand of peptides to develop this helix. The alternative to an alpha helix is a beta-pleated sheet, and we see this in lots of proteins like GFP, which is green fluorescent protein, and also in silk. So again, we're going to see this hydrogen bond form between the slightly positive hydrogen that's part of the amine terminal and the slightly negative oxygen that's part of the carboxyl terminal. So the primary structure of the protein is based on the sequence of amino acids, and then the secondary structure is based on hydrogen bonding between the carboxyl terminal of one amino acid and the amine terminal of another amino acid. But then the really interesting complex 3D shape, which we call the tertiary structure, is based on the interaction between the different R groups of the different amino acids. So there are four main ways that these can interact. There are hydrogen bonds. There are disulfide bridges or disulfide bonds, which are going to occur between sulfur-containing R groups, in other words, cysteine. Hydrophobic interactions between non-polar R groups, and then also ionic bonds between oppositely charged R groups. This can lead the protein to be further coiled or folded, and it determines the shape of really complex proteins, particularly things like the active site of enzymes. So when an enzyme becomes denatured by extremes of temperature or pH, it's this 3D structure, this tertiary structure, that's being lost. So where we've got amino acids that have oxygen in their R groups, it's possible for us to see this slight delta positive and delta negative arising, and this can lead to a hydrogen bond forming between these. So this is comparatively weak compared to the disulfide bridge that we're about to look at, but it can still change the shape of the protein. And then where we have cysteine R groups, these contain sulfur, and it's possible for these to bond together and form a covalent bond that we call a disulfide bridge or a disulfide bond, and these are incredibly strong bonds that are going to pull two bits of the protein together. And then our third interaction is where we have hydrophobic R groups. So these are non-polar, and basically these are going to clump together to try to stay away from any polar groups or any water. Now, for lots of proteins, 3 is enough, and all they have is a tertiary structure. But for many complex proteins, you have multiple subunits joined together. And this happens in fibrous proteins like collagen, globular proteins like insulin, and also conjugated proteins like hemoglobin. And conjugated means that they have additional components that, strictly speaking, aren't protein at all, but they're part of that thing that needs to work. So where you have these different subunits, you could have two subunits that are the same. So for instance, lots of enzymes, that's true. You can have two different subunits, which is the case in insulin, or you could have a situation where you have both. So hemoglobin has two alpha subunits, which are the same as each other, and two beta subunits, which are the same as each other, and also these 4 heme groups containing iron 2+ ions. So just before we finish, can you describe what is meant when we're talking about protein and we're talking about the primary, secondary, tertiary and quaternary structure? So the primary structure is just the sequence of the amino acids. The secondary structure is where we then have hydrogen bonding, turning this into either an alpha helix or a beta-pleated sheet. The tertiary structure is a 3D structure that is caused by hydrogen bonding disulfide bridges, hydrophobic interactions and ionic bonds between the R groups, and a quaternary structure is where we then have 2 or more polypeptide chains. Thank you very much for watching, and I hope you found this video useful. If you're interested to learn more about globular proteins and fibrous proteins, then that's the next video in the playlist. If you did find this useful, then don't forget to like and subscribe, and I'll be back soon with some more A-level biology videos.